Please welcome Francisco Buchara, SF500.
Hi, I'm Francisco Buchara, CEO and Managing Partner at SF500, a Nobel Science Fund and company building based in Rosario, Argentina. We are just starting, and you know what? ONCOLIQ is the first one of 500 startups that we want to support in the next decade. And it's not a coincidence. ONCOLIQ has everything we look for in a startup: deep science, a brave and community-oriented teams, and of course, global impact. We have nominated ONCOLIQ for its strong cancer and microRNA science, which has demonstrated great results in early cancer detection clinical studies with a sensitivity for breast cancer of 90%. Finally, I would like to add that Adriana and Marina, the two co-founders, are two amazing people that have shown tremendous energy, determination, and drive to make things happen. Thank you, everyone, and have a wonderful night celebrating science. Ciao.
Breaking the wall to early cancer detection. Marina Simian, ONCOLIQ.
Hi, nice to meet you. So COVID will be remembered as a stepping stone that led to universal early cancer detection. And why is this? Because COVID led to the decentralization of PCR testing. Today we have PCR machines everywhere, here in Berlin and in a small town in Latin America. I am going to show you today how ONCOLIQ is developing its technology using PCR machines and how they are a central part of our business model. So the mission of ONCOLIQ is to reduce global death by cancer. Between 40 and 50% of adults will have cancer at some point in their lifetime. And with current screening technologies, half of these people receive a late cancer detection, meaning that most of them will die within 5 years. Additionally, 71% of deaths by cancer are due to the development of tumors in organs that do not have a method of screening. Our solution is a multi-cancer detection early blood test. What we do is combine molecular biology based on PCR to detect circulating microRNAs with machine learning, and thus develop algorithms that allow us to determine early on if a person has a tumor in a specific organ. Our R&D process consists of the following steps. First, we identify a set of candidate microRNAs that are biomarkers for each type of cancer in circulation. We then validate these candidates, and with the information that we generate, we produce the machine learning algorithms. Finally, we pilot test the technology in a real-world scenario. We are at this point developing the technology for 5 types of cancer. The most advanced is the breast cancer technology that is currently being pilot tested in Argentina, and we hope to go to market next year in Latin America. Our business model is decentralized. We will license the manufacturing and distribution of PCR kits that should reach every corner of the world, so every clinical lab that has a PCR machine can offer an ONCOLIQ. We will provide a data processing service in the cloud, produce the reports that we will deliver, and charge a fee for service. Today, there is only one company providing this type of service, a blood test for the detection of 50 types of cancer. It is called GRAIL, and the product is Gallery. Gallery's sensitivity for 50 types of cancer is 50%. This means that if they test 100 people who have cancer, they only detect 50. Gallery's sensitivity for breast cancer detection is 30%. ONCOLIQ Breast, our first technology, has a sensitivity of 90%. The size of the market, considering only all of America and Europe, adults over 40 and a price of ONCOLIQ of $100, is of $73.1 billion. So far, we have established collaboration with 19 medical institutions, 43 physicians, 2 local governments, and 1 health insurance company. We have a biobank with over 2,000 patient samples as a result of 33 clinical trials. We have launched our breast and prostate cancer pilot studies. We already have our first patent that was published in the US in September. We have reached an agreement with the largest CLIA certified lab in Argentina, and we have launched the development of our cloud platform. We have received so far $1.1 million in investment and grants. We will open our seed one in March 2024, looking for $5 million. This will allow us to discover and patent the biomarkers for 30 types of cancer, execute our business development plan, and go to market and launch sales. This company was founded by Dr. Adriana de Siervi and myself. We both have more than 20 years of experience in cancer research. These are our current advisors and investments. We invite you to join us to reduce death by cancer worldwide with a technology that is decentralized, low cost, and highly sensitive. Thank you very much.
Wonderful. I wish I would have seen you here on stage 5 years ago, because maybe my best friend would live today. Thank you. So let's go, yes, on fight against this. Please, Chance first, and then we will have, okay, first, yes, you go.
Can I ask an ignorant question? I mean, I know there's lots of labs across the world trying to do this. So what's your edge? What's your USP?
Our edge is our expertise with the microRNAs. So this actually started as an academic project in 2014 in mice, when very few people were working with microRNAs. It started in a breast cancer model, and then it went to the blood of the mice, and then it went to the blood of the patients. So it's really been 7 years of R&D in academia. And in 2021, Adriana, my partner, who is the one who developed the technology, she called me and she said, Marina, we have to do a company with this. We were collaborating on the project. And basically using microRNAs, there are very few companies. And what I see is that the sensitivity of the circulating tumor DNA is not high, because tumor DNA goes into circulation much later in tumor development. So if you look at the different stages of development, for example, GRAIL has high sensitivity at stage 4, but then it's too late. I mean, it doesn't make any sense. So microRNAs go up in circulation very early on. We still don't know why. I mean, this is not, we just use them as biomarkers.
Thank you. Rudolf was the second one, please, in the first row here in this block. Here you go.
Yeah, thanks a lot for the presentation. I mean, driving sensitivity up helps to avoid false negative. On the other hand, the other side of the story is the false positives. What about the specificity?
Yeah, our specificity currently is in 80%, okay? But, well, we are actually working on our machine learning algorithms and trying different strategies to improve this. But we think 80% is pretty good. We are more worried about the false negatives. I mean, every technology has false positives and false negatives. For example, a mammogram has a sensitivity of 85% average, but if you have a dense breast, then it's 60%. So what we have today isn't perfect. And I really think that we have to, try to incorporate new technologies to save more lives and detect cancer early, basically.
Thank you. I just checked the jury doesn't have any question right now. Angelica, yeah. Sorry, yes.
No.
Yes.
Thank you very much for giving me the opportunity for the question. I have one question for the commercialization. Do you plan it as preventive diagnosticum?
Well, it's not preventive because you have the cancer, okay? But if you detect early, look at skin cancer. You have something very small, you remove it because you see it. We cannot see our organs. Well, let's go and look at them if we have an indication that something is going on.
And what do you think about the clinical trial you need? I mean, you need a lot of data to validate this product.
So currently, the breast cancer pilot study, we already have 640 ladies who have gone to their annual checkup. They have given us a sample of blood and their mammogram. We are aiming for 1,000 in this first study. And we are having the mammograms analyzed by another startup in the US that has a technology to look deeper into imaging, because we think, as ONCOLIQ can detect very early, that there will be cases where the mammogram, the way it is analyzed normally, or the woman, or the lady has a dense breast, will be negative. And we will have a positive result. So actually, we're working on that.
Okay. Is this a yes-no question? No. Okay, no. In 13 seconds, I cannot take any new question with yes, no. If you have one question with 2 words and a yes, no answer, yes. Otherwise, I think we are
Thank you
done.
very much.
Thank you so much.
Thank you.
Thank you.